resta in tasca? Sì, bravissimo. Metto il timer. Perfetto. Okay, good afternoon, everyone. So today talks has a very long title because I want to present you something scientific, but also a novel tool that we developed. And all the work has been done together with Dr. Thomas Bose and Professor James Marshall at the University of Sheffield within the ERC project Diet. And the main topic is about psychophysical laws. And psychophysical laws explain the relationship between stimulus intensity and its perception in the human brain. And this type of relationship has been observed for a large variety of stimuli. And later, scientists observed that not only humans respond to this type of psychophysical laws, but has been observed also in mammals, fish, birds, insects, and more recently, scientists noticed that also individual cells like the slime mold might follow this type of relationship. So this is particularly interesting because we can observe these psychophysical laws also in absence of a brain, so in various type of organisms. And what we do here is to check if another type of organism follow these psychophysical laws. And the organism that I will watch is the superorganism. And for superorganism, we refer to the colony of insects. So if we think about the insect as the organism, the superorganism is the collection of them that they work together. And if we watch at the colony as a unique entity and how information spread within the colony, then we can start to draw parallels between the superorganism and how information spread in the brain and between neurons. And this parallel has been already observed and described in various work. We are not the first, but we bring ahead this idea and we follow this line of research. And in our case, we investigate the psychophysical laws, as I said. And as reference model, we use the honeybee nest site selection. And this model explains how honeybees select their future nest. This process happens in spring in European bees. And when the colony reproduces, the old queen leaves the nest, goes to a temporary place together with some thousands of scout bees. And the bees will need to find quickly the future nest location. And they are exposed to danger, so they need to find quickly a good solution and also a consensus solution. They don't need to split. And how they do that, scout bees will explore the environment and might discover potential nest sites in the environment. And bees that discover a place, a good potential place, will return to the swarm and will actively recruit other scout bees through the woggle dance. Then, when two bees committed to two different nest sites encounter with each other, one delivers stop signal to the other, and the one that receives stop signals will revert to an uncommitted state. Finally, bees will spontaneously abandon commitment for poor quality nest sites. So from field observation, biologists observe that the bees manage to make this collective decision through these four types of individual bee actions. But how the colony selects the best option? I can explain you this through an example in which we have two nests, the blue and the red, and we label as green the uncommitted bees. And we can write this for action in a more formal way in a chemical reaction-like form. So if we have an initial state, uncommitted, and a final state that is commitment for the blue, and that happens with a certain rate. And so on happens for the red nest, and upon encounter of a blue and a green with a certain rate, the blue will recruit the green, and so on for cross-inhibition and abandonment. And bees, when they before getting committed, they visit the nest and estimate the quality of the nest that we can label as V1 and V2 for the two nests. And the key idea is that the bees will modulate their action as a function of the estimated quality. So they will do more frequent discovery, recruitment, cross-inhibition for better nests. And that will steer the system toward the convergence, toward the best option. So the idea here is to parameterize, let's say, write all this rate as a function of the quality Vi. This is the parameterization proposed, in which everything is dependent on Vi, and we introduce two terms, the term K that modulate the frequency of individual action, and the term H that modulate the frequency of signaling. So we can study our system in this ratio of signaling over individual behavior with a single parameter. And so what we do is to analyze this model. This work is a theoretical work. We just analyze the model that comes from field observation to see disagreement with psychophysical laws. And we do the analysis of the model in the form of the master equation that includes stochastic fluctuations. And here I pause a little bit the presentation because I will show you the tool that we developed that allow us to do this type of analysis. So, the tool is called MUMOT, Multi-Scale Modeling Tool, developed in our group. And it is written in Jupyter, that is a Python, Python framework. And Jupyter allows you to write notebooks. And they are somehow similar to mathematical notebooks in which there are cells that can be dynamically run, and you can interact with the cells, with the various cells with the commands. And it's presented as a website that you can run in your browser. Yeah, for lack of time, I will not run online the cells, but it's already a pre-run notebook that anyway show the main functionalities. So here we just loaded the library, and here the idea is that we want to write a model that in this form of chemical reaction. And in fact, to make it bigger, James. I don't know how to make it bigger. Okay. So the tool allows us to write and analyze any type of model that can be written in this form, in which there is an initial state, a final state, and a constant transition rate. And this is particularly useful because usually biologists, field experimentalists, often build up their model in this form. They see a change of state with a rate, and so they can take the model derived from observation and put inside this tool and make a first analysis of it. So here we write the model with the specific syntax, and here we parse it. And here is visualizing which we visualize the transition in which we label A and B as the two possible nests and U the uncommitted bees. And then once we parse the model, we can show the ODEs. That is done automatically. We have the ODE system that we can then integrate over time. Here there is a time integration of the ODE, and there are widgets that allow us to modify the parameter and have real-time the change of the system. So with widget, you can really play with the model and have a feeling of how things change. And then you can also visualize a vector field that show you in a bidimensional space how the trajectory vary by varying the parameter. Again, with widget, you can vary it real time. Then we can visualize also a stream plot with a stability point or unstable fixed point, the hollow in the middle. Or we can also run a bifurcation analysis. Here, for example, we see that by increasing the value H, that was our signaling, we have a pitchfork bifurcation. And then the interesting part for this study is the SSA, that is the stochastic simulation algorithm proposed by Gillespie that allow us to find the approximation of the master equation. And as you see here is a stochastic run in which include fluctuation due to the finite system size. And if we reduce the system size, we have larger fluctuation, and if we increase it, the fluctuation are much smaller, as expected. And then we can use this type of finite system fluctuation to estimate which is the noise around the stable point here for the system size 70. And then finally, you can also visualize a multi-agent simulation results in which agents are the dots that communicate on a fixed topology that is indicated by lines connecting the agents. And this is a fixed topology simulation, but we can also have a time-varying topology in which particles move in the space and only interact locally with the neighbors. So this is just a quick overview. If you have more questions, you can find me later and ask more. So going back to the main presentation, we use this tool to approximate the solution of the master equation through the stochastic simulation algorithm. And we did so for studying if our superorganism respond to psychophysical laws, as is in agreement with them. So we systematically vary the decision condition, quality, and number of potential nest size to study how they relate to the ability to discriminate, so to select the best quality and location, and how much time they take to make the decision. And we do that to study these three laws, the Weber, the Pierron, and the Hicks-Hymans law. I want to mention that has been already observed in insects. Something happened. Yeah, try to probably disconnect. Okay, we are back. So yeah, I was just mentioning, I was spoiling, that's why he went away. It was a spoiler, that, because probably next presentation will tell us that has been already observed that insect might follow psychophysical law, but the study investigated at make the study at the level of the single individual. Instead here, we don't watch single individual response, but we study the things at the level of the superorganism. So now I will go through these three laws. I explain you what they are and if we find an agreement. Let's start from the Weber's law. Weber's law describes the relationship between the stimulus intensity and its discriminability. So I explain you what it is through an example on numerosity. So now you have to tell me which is the larger set that you observe. So I will present you two sets, you need to reply. So this is set 1, and this is the second set. So please raise your hand if you believe this is the larger set. And now raise your hand if you believe this is the larger set. Okay, everybody got it right. This is the larger set. Now again, a second experiment in which you have again to find the larger set. And these are the two sets. So please raise your hand if you believe this is the larger set. And now please raise your hand if you believe this is the larger set. Okay, still you did pretty well, but there were many more errors. Why there were many more errors and you couldn't, not everybody could reliably distinguish the same difference? Because the difference was still 5, but 5 is enough to correctly discriminate between 25 and 30, but it's not enough to correctly discriminate when we have larger set. This is explained by the Weber's law that say that the minimum difference between two stimuli that an organism can correctly discriminate is a constant fraction of the base stimulus strength. So we can write formally like this, or if we have on the horizontal axis, the stimulus strength, so the number of dots, the just notable difference increase linearly. That is what we expect to see. And in our system, we varied the option quality, so the quality of the nest location presented to the swarm, and we monitor the decision outcome when they reliably, so the 75% of the time, select the best one. And what we find is indeed a linear relationship. And also what we notice is that the accuracy, the ability to discriminate between two sources improve, it becomes more precise with the increase of the swarm size. This is due to the random fluctuation that we saw before playing with the tool, in which smaller swarm have larger fluctuation and leads to larger number of errors. Then we move to analyze the Pierron's law, which studied the relationship between the stimulus intensity and the reaction time. And again, I will explain what it is through an example, this time on brightness. So you have to see when the circle appears, and will appear here at the fixation cross. And then, it appeared, and then a second circle will appear again below. And what has been observed is that you are slower to react to this circle and quicker to this one. So the brighter, the quicker. This is explained by the Pierron's law that says that the mean reaction time decreases as a power law with increased stimulant strength. And we can be formally written like this. If we have on the horizontal axis the stimulus strength, what we expect is a power law decrease. And in our case, what we did, we varied the option quality, and we monitor the time to make the decision. So this is the data from the experiment, the simulation data, and they nicely fit with the power law. Then finally, we moved to investigate the J.A.R. law, in which we studied the relationship between the amount of information and the reaction time. And I explain again quickly through an example on what is on brightness, what is the J.A.R. law, so which is the brightest circle you have to locate between three options that will appear here. And the second example is instead on 6 options, again to find the brightest circle. And the idea here is that you are quicker when there are 3 options and slower when there are 6. And this can be described by the fact that the reaction time increases with the amount of information to be processed and can be formally written in this way, where S is the time to process 1 bit, and I is a function of the number of alternatives. And the function can have various forms, and that depends on the type of task. And in literature, we found that theoretical study on neurological model observed that for this type of task, value-sensitive decision in best of n case, there is a non-linear increase. So that is what we expect to find. So what we did is to increase the number of possible nests and to measure the time to reach a quorum. This is the data that we obtain, that they fit with an exponential curve. This might be scary because you say the system doesn't scale well, although we also notice that the swarm can counteract this fact by increasing signaling. So by speaking more, transferring more information, there is a power law decrease. So somehow the swarm might balance this exponential increase by increasing the communication. So with this, I conclude. I just want to mention as first thing that this is a theoretical study. We didn't work with real bees, but comes from a model that is derived from field observation. And what we observe is that the emergent behavior of the superorganism may obey to through these 3 laws. I put in red and bold the word emergent because what we see is an emergent property, because none of the single individual follow anything that is linked to these psychophysical laws. But what we observe is that the group, the superorganisms display this relationship. So we are in front of a form of swarm cognition. And this is part of a larger project, as I mentioned, led by James Marshall, the Diode project, in which we try to find links between various systems and find the common motifs that explain this system. This is a step forward in this direction, and in which we link the functioning of the brain to the superorganism that is composed, instead of neurons, by insects. With this, I conclude. Thanks for your attention.